So this is digestive system, part 2. We're going to cover the intestines in this PowerPoint. The first organ we're going to discuss is small intestine. And just looking at this image, you can see both small intestine and large intestine, the difference between the two being a diameter issue. The large intestine also happens to be shorter at around 5 feet, that measurement taken in a cadaver, while the small intestine measures around 20 feet, again, that measurement taken in a cadaver. The three main regions here are the duodenum or duodenum that connects to the stomach by way of a pyloric sphincter. That's about the first 10 inches of the small intestine. That's actually where the most absorption occurs. The second region or middle region of the small intestine is called the jejunum. That accounts for about the middle 8 feet of the small intestine. And the ileum is the terminal or last portion of the small intestine, and that is the last 12 feet. The small intestine connects to the large intestine approximately in this area here, and there's another valve there that regulates the passage of food, or what's becoming waste now, into the large intestine. And that ileocecal sphincter or valve is located between those two organs. So let's talk about 3 modifications inside the small intestine to help increase surface area. If you had to remember one word to describe this organ's job, it is absorption. So these 3 modifications all help to increase the surface area so we can increase absorption of nutrients into the bloodstream. The first are finger-like extensions known as villi. The second here are called microvilli, and the third are called circular folds. So I'm going to take advantage of the white space on this PowerPoint slide and show you what these 3 look like. When you look inside the small intestine, some of this stuff is visible with the naked eye, and then of course some of it is microscopic and you can't see it. So let's start by talking about villi and microvilli. A villus is a finger-like extension. So up here I'm just going to draw a finger-like extension of the small intestine lining. Okay, so each one of these little extensions would be called a villus. Plural, they are called villi. And the lining here is simple columnar epithelial tissue. And then, of course, that makes the lining as it goes up and back down, just like that. Now, inside each villus, you're going to find a capillary bed. You're going to find lymphatic vessels called lacteals. But let's take that villus and look more closely at it. If you were to look at an individual villus, you would notice that it had little teeny tiny extensions like this called microvilli. So it kind of looks like a Christmas tree or a pine cone. So that's when we take this single villus here and look at it in more detail. Each one of these extensions here is called a microvillus, or plural, they're called microvilli. All right, so when we're talking about circular folds are groups of villi. So I think I'll show you a picture so you can better see how they work. Okay, once again, there's a villus, capillary bed inside, simple columnar epithelium. Okay, more villi. They kind of look like a bumpy surface of a tongue. And then nice, great picture of those microvilli. This right here is a single villus. There's another single villus. All right, great picture here of circular folds or plicae circulaire. You can see that it looks kind of like a speed bump. If you take one plicae circulaire or circular fold, let's just circle one of these villi. Right there is one. And so if we were to pull up the magnification, we would see that there are individual microvilli all over that villus. Now the point of the plicae circulaire is to slow the passage of food through the digestive tract. This gives the small intestine more opportunity to absorb fluids and food nutrients into the bloodstream. So I kind of think of these plicae circulaire as speed bumps. Okay, so in summary, we've got 3 modifications to the small intestine lining. They help increase absorption and the opportunity to mix enzymes with food. And so from the smallest structure, they are microvillus. Microvilli are found on villi. They help to form these circular folds or plicae circulaire. All right, so there you can see kind of that bumpy appearance inside the small intestine lining. Just a side note here, celiac disease destroys the surface area inside the small intestine. So what impact do you think that will have on our ability to absorb if celiac disease destroys those villi? Decreased, right? All right, on your worksheet here, about middle of the way down, page 88, you'll notice there's around 1 to 2 liters of intestinal juice secreted into the small intestine, and the pH here is not acidic. It is going to be slightly alkaline or basic, around 7.4 to 7.8. So we're going to talk about why the environment here becomes alkaline when it was so very strongly acidic in the stomach. All right, so very bottom of page 88, you'll notice there is a summary of activities here. Food spends around 3 to 6 hours moving through the small intestine. That's booking it when you're talking about approximately 20 feet of intestine that we're moving through. So here in the small intestine, we're looking at some mechanical digestion through peristalsis and segmentation. There is a lot of chemical digestion. In fact, this is where we finish the process of breaking down all food groups. And all nutrients that are going to be absorbed are done so here through the wall of the small intestine. So let me show you segmentation. This is a specialized type of movement that occurs only in the small intestine, and I would think of it kind of like a washing machine effect. You may see the colors here in each portion or segment of the small intestine, and they are mixed back and forth with one another. The whole goal of this back and forth movement is to help mix enzymes and food groups together. That increases the ability to break down those food groups. Again, what is the whole main goal of the small intestine? It is to absorb those nutrients into the bloodstream. All right, so segmentation is a back and forth movement, although peristalsis is still moving the food toward the ileocecal sphincter so it can be pushed into the large intestine. This is a good time to talk about food groups and fat for just a moment. So we have talked about carb digestion in the mouth helped along by salivary amylase. We talked about protein chemical digestion in the stomach assisted by pepsin and renin. We have not yet had any discussion about how fats are digested or nucleic acids. We'll hold on the nucleic acids for just a few moments, but let's talk about fat first. What you're looking at is a portion of the small intestine. You'll notice it's called the duodenum or duodenum. And there's a connection here from the pancreas, liver, and gallbladder. We're going to focus on the gallbladder because that gallbladder stores bile and the liver produces the bile. So when food comes into the small intestine and travels through here, there are hormones, we'll talk about in a few minutes, that travel through the bloodstream. They go back and they target both the gallbladder and the liver, which is sitting up here, to release bile into the small intestine. So if you've ever seen commercials for Dawn dishwashing liquid, they advertise themselves as being the product that helps to break up grease. So that means that big globs of grease using Dawn dishwashing liquid are broken into smaller, more manageable globs of grease. And that's exactly what bile does. We call it an emulsifier. That means it takes large blobs or globs of grease and breaks them apart into smaller, more manageable pieces. Let me emphasize that bile is not an enzyme. Bile is kind of like a detergent. It's an emulsifier. So you've got to have the bile present in the small intestine in order to start breaking apart big globs of grease or fats into smaller, more manageable ones. Then we also use enzymes, lipase for one, to help break apart or hydrolyze those fatty molecules into their building blocks. You may recall the building blocks of fats are fatty acids and glycerols. So just to review here, the gallbladder stores bile, the liver produces it, but when you've eaten food, both the liver and gallbladder secrete their bile down through this common bile duct, joins here with this main pancreatic duct. We'll talk more about that later. And then, of course, the bile here is moving through that hepatopancreatic sphincter into the small intestine. And the whole point of bile is to handle big globs of fat and emulsify or break them apart into smaller, more manageable globs of fat. You got to have enzymes that also play a role. It's kind of like a 1-2 punch. All right, so we're breaking down all food groups here. You'll notice near the bottom of your page, pancreatic enzymes and what's called brush border enzymes are present inside the small intestine. They help to break down all the other food groups. That is, proteins, carbs, lipids, once again, and nucleic acids need those enzymes that come from the pancreas and the small intestine itself. Finally, on the very bottom of 88, the main goal here for the small intestine is absorption. That means food groups, some water, and electrolytes are going to be absorbed here. All right, so the reason absorption can occur here so easily is because we're talking about a single cell layer between the food in your intestines and the capillary bed. So we've got a very thin layer of simple columnar epithelial tissue, and inside the villus you've got a capillary network and a very small lymphatic vessel called a lacteal. The reason it's called a lacteal is because some of the fluids that move in there are milky in color, kind of a white milky liquid, and that milk color is where the lact comes from in the lacteal term. All right, so if you will follow me over to page 89, we're going to now talk about specific absorption in the small intestine. This information out of anything in this chapter is great for flashcards. You'll want to be able to keep track of where digestion for a particular food group begins and where it's completed. And make this really easy on you because all food groups are completed in their digestive process, meaning chemical and mechanical digestion here in the small intestine. So with the carbs, the breakdown process begins in the mouth using salivary amylase. Like we talked earlier, that is not a complete process. You would actually have to hold a piece of starchy bread in your mouth for like 20 minutes in order for amylase to complete its job. So when starches come to your stomach, the stomach just breaks them down mechanically through peristalsis. But here in the small intestine, both segmentation and peristalsis grind the food apart, but it's these enzymes listed here that help to break down those starches and sugars into their building blocks. Notice that a lot of these enzymes do come from the pancreas, like pancreatic amylase. Some of these enzymes are native to the small intestine. So just a great review here, that ASE prefix means enzyme, and of course the prefix there tells you what we're breaking down. So lactase breaks down lactose. That, of course, is a milk sugar. Some people are lactose intolerant because they do not have enough lactase. Sucrase breaks down sucrose. Maltase breaks down maltose, another sugar. And amylase, of course, breaks down starch. Protein digestion also is completed here, but keep in mind, chemical digestion of proteins begins in the stomach. We can do mechanical digestion by chewing in the mouth. Peristalsis can help break these molecules apart, but the large-scale proteins apart. But chemical digestion here means we're hydrolyzing. That means we take proteins and break them apart into their amino acid building blocks. So pepsin and renin both help to chemically digest proteins into amino acids in the stomach. That process is not completed, though, until you reach the small intestine. The pancreas releases these enzymes here, trypsin and chymotrypsin, into the small intestine. That helps to assist and complete the process of protein hydrolysis. So just as a way of review, earlier you probably learned that beads on a string is how these proteins were built in a primary structure. These enzymes come along and break those proteins apart. And so what we're left with are individual amino acids. Another helpful hint, notice the syn suffix, trypsin, chymotrypsin, even pepsin. Hey, renin's close enough, right? So these syn suffix words are ones that break down or hydrolyze proteins. All right, so that breaks them into their building blocks or amino acids. Lipid digestion in a very small amount begins in the mouth using a little bit of lipase. That's why fatty foods sometimes have a different feel to them. Again, that ASE is an enzyme, lip being lipids there or fats. Now, There's no chemical digestion of lipids in the stomach, but when we get to the small intestine, we are looking at two things happening. First, we have to have bile present in the small intestine to act as an emulsifier, and then we need lipase there to act as an enzyme that chemically breaks apart the lipids into their building blocks, which are fatty acids and glycerols. So imagine if you've got a large piece of fat that comes into the small intestine. Bile breaks that up into two. All right, and then keep breaking it up so we now have more manageable pieces. And then lipase comes along, and lipase will break down these molecules into their fatty acid chains and their glycerol heads. So if you have only lipase, it just can't manage on a really big piece of fat like you see here to begin with. So that brings me to this story. If you happen to have not enough bile in your small intestine to emulsify fats, then the fats will be unprocessed, not absorbed, and they'll leave in the stool. So people who have poop that floats end up having too much fat, and that may be an indicator that bile is not able to break down those molecules or break down those globs like they need to. Likewise, bile gives stool a particular color, kind of a brownish color, and that is due to the breakdown of the pigments that are present in bile. So if someone has light-colored stools, beige-colored stools, then that also could be an indicator for something going on in the liver, gallbladder, and their connection to the small intestine. So floating poop, consistently floating poop, means it's full of fat. And then, of course, light-colored stool means that bile is not making its way into the small intestine. That, too, could be a warning sign. Now, little kids who drink nothing but milk may have light-colored stools, and that's normal for that age group. But, you know, once people start having a normal, regular diet, then we expect to see brown-colored poop. All right, that's enough for poop today, all right? All right, so we are now talking about nucleic acids. We haven't really discussed them at all, but these nucleic acids come from the organisms that we eat. You know, the old saying goes, you are what you eat. So you eat corn, you get corn DNA. You eat cow, you get cow DNA. And your body takes these DNA and RNA nucleic acids and breaks them down using nucleases. That should sound familiar, right? ASE, of course, being an enzyme, and nuclease sounds like a nucleic acid, into their building blocks. Things like adenine, thymine, what are the other two? Guanine, cytosine, and even uracil. So we use those building blocks, those bases, to help make our own DNA, RNA, or even ATP molecules. Finally, you know, we're talking absorption here, so when we eat foods, of course, we may be eating vitamin-rich foods, and we will absorb vitamins into our bloodstream through the wall of the small intestine. ADEK, A, D, E, and K are fat-soluble vitamins. They have to be taken with food to maximize their absorption into the small intestine's capillaries. B and C are water-soluble, and so they can be taken on an empty stomach, or they don't necessarily need to have fat in your system to be absorbed. We actually make Burger King, B and K, in the large intestine. We'll talk more about that when we move to that organ in just a little bit. And if you flip on over to page 90 with me, we will finish up with electrolytes. We absorb iron and calcium in the small intestine if needed. We absorb sodium and we couple it with glucose and amino acids. Potassium is absorbed by facilitated diffusion. So that brings me to the next question. If you don't absorb these electrolytes, your blood osmotic pressure is going to change. Will it increase or will it decrease? It will decrease. So if you think about it, somebody who has perhaps had part of their small intestine removed, perhaps had a disease that damages the lining of their small intestine, may end up having problems reabsorbing fluids back into the bloodstream, because that is a job of blood osmotic pressure. So hold on to that thought, because we will come back and revisit this conversation when we talk about fluid electrolyte balance later on in this course. Finally, Water is absorbed in the small intestine from what you drink, and then we're secreting saliva constantly, stomach juices, and intestinal fluids. So of about 10 liters or so of fluid that come through to the small intestine, we are reabsorbing about 9 liters. That's about 90% of what we're absorbing. The remainder will be absorbed through your large intestine's lining. So this is a lot of fluid per day that's moving through your digestive tract. And notice here, less than 1% of what you are excreting in your feces is actually all that's left. We're reabsorbing almost all these fluids back into our bloodstream. All right, so that concludes the small intestine. The number one job here is absorption of all food groups and the complete digestion of all food groups here.